Hello everyone, welcome to the new session of Mendel Laws of Inheritance. So this is the first part. Okay, so let us start this session. Okay, so what does the Mendel laws says? Okay, though the laws of inheritance were derived by Gregor Mendel. So this of inheritance was derived by Gregor Mendel in 19th century monk conducting hybridization experiments in a garden piece. So he was a monk in a church and he used garden pea, that is a Pisum sativum, for his experiment. Okay, and he conducted hybridization process for his experiment, right? Between 1856 and 1863, he cultivated and tested some 29,000 pea plants. So by profession, he was not a biologist, not a botanist. Okay, but still, due to the curiosity, okay, he was using this plant, that is a garden pea, for his experiment. So in 1856 and 1863, he cultivated garden pea plant and tested some around 29,000 plants. Okay, from his experiment, he deduced two generalization, which later becomes to know as Mendel laws of heredity or Mendel inheritance laws. Okay, Mendel discovered that by crossing white flower and purple flower, the result was not a hybrid offspring. Okay, so he took white color flower and purple color flower of the pea plant and he got the result that was not a hybrid offspring. Rather than by mix of two, the offspring was a purple flower. So he was confused that what occur for this white flower, where does this white color gone, why I'm not getting a light purple color flower or a mixed flower or a white flower, why I'm getting only purple colored flower, right? So he then conceived the idea of hereditary units, which he called a factor. So by having a lot of such hybridization result, he concluded some points. And what are those? That there is some hereditary unit that is transferring from one generation to the another. And to that hereditary unit, he gave a term that is called as factors. So Mendel said that factors, later called genes, the factors which was given by the term by Mendel was later came to know as gene, normally occur in pair in ordinary body cells. So normally they are genes which are present on the chromosome. Suppose I'm showing a gene. So this is a pea plant. This is a pea plant which is having a chromosome, and for the tallness they are having a gene at this particular locus. This is for tallness. So one, this is capital T and this is showing T. So I am showing capital T for the tallness, of this particular pea plant. So this is he called gene. So I am showing that this particular gene for the tallness, he gave a name that is called as factor. Because at that particular time, there was no idea of chromosome, no idea of gene, that at chromosome there is a locus for each gene. Each character is represented by a gene in a cell. So there was no idea, but still he concluded that there must be something which he called as a hereditary unit, and he termed that as a factor. Later, that same factor came to know as gene. Okay, that occur in a paired form in an ordinary body cells, and yet segregate during the formation of the sex cells. So when they form the sex cells, this will get T and this will also get T. And these two chromosomes get segregated at the time of gamete formation or the sex cells formation, right? So the dominant gene, such as the purple flower in the Mendel's plant, will hide the recessive gene, the white flower. So one of the factor or one of the gene for the same character was a dominant. What is meaning of the dominant? That dominate, the one which dominate the other. And the recessive means that gets suppressed by the dominant one, right? So this capital T, this is the purple flower. So I'm taking the example of flower. Okay. So I am taking this capital P for purple. This is the purple flower and this is the white flower. Purple flower and this is the white flower. So this will be taken as small p, not W. It will be taken as small p because it is depend upon the character. So we are taking the flower color as a character, flower color as a character, and at a locus of the chromosome, this purple flower will have capital P, and this white flower will have at the same locus small p, small p. Now what happened? The gamete will be formed, that will be capital P, and here the gamete will be formed as small p, right? So what happened? So there will be fertilization. So here we can have four combination. So this will have capital P, small p, capital P, small p, capital P, small p, and again capital P and small p. So all the types of offsprings are capital P and small p. But the flower appear as purple. This flower appear as purple. Why so? Because this capital P is a dominant gene or a dominant factor and it suppress small p. So small p is not able to express itself. So small p will not be able to express itself in the presence of capital P and the flower appears as purple only. Okay, so Mendel started that each individual has two factors for each trait. This we have already discussed, that for each individual has two factors for each trait or character. This trait is also known as character. For example, tallness, flower color, right? So these are the trait or the character. And for this tallness and flower color, we will have two factors, right? For tallness, let us say for tallness, capital T for tall and dwarf small t. So two factors. Now flower color. Just now we have seen in the example, capital P purple and small p white, right? The two factors may or may not contain the same information. Okay, so they can have in this form capital P, small p, or they may have capital P, small p. So this will be pure purple, this will be hybrid purple, and this will be white. Okay, so the two factors may or may not contain the same information. So this factor, this paired factor, and this paired factor may or may not contain the same information, right? So if the two factors are identical, then it is called homozygous. Then it is called homozygous. Suppose we are taking for the tallness, height of the plant, capital T, then it is homozygous tall. And if it is having small t, then it will be called as homozygous dwarf. Right? So like this, we can have the example of flower color also. So capital P or small p. Homozygous. Okay. This is also the homozygous condition. If the two factors have the different information, the individual is called as the heterozygous. So if it is having capital P, small p, then it will be heterozygous. As we have seen in the one of the cross of the purple and white flower, right? If we are taking the example of tallness, then it can be capital T and small t. Again, it is heterozygous. Okay, so the alternative form of a factor is called as allele. Now, this is very, very important. For one character or a trait, we can have number of alleles, generally 2, but may have more also. So if we are taking tallness, then we are having capital T and small t. So this is the allele. Okay, if we are having flower color, then we will have capital P and small p, right? So this is for purple and this is for white. So these are the alleles. The genotype of an individual is made up of many alleles it possesses. Okay, so the genotype, the one which we can see by the paired chromosomes and a concerned allele, that is capital P, small p. So this is showing the genotype, right? And how it appears, the plant appears morphologically, that will give you the phenotypic character. So this will be a hybrid purple. And capital P, this will be again a purple, that will be pure purple flower. Okay, so here this one, this capital P, small p, this is showing the genotype and how it appears morphologically by the appearance. Pure purple or hybrid purple, this is the phenotype. Okay, so an individual physical appearance or the phenotype is determined by its allele as well as its environment. So two things are there for the phenotype: the allele combination as well as the environment. Okay, so an individual possesses 2 alleles for each trait. One allele is given by the female and another comes from the male parent. Alright, so they are passed on when an individual matures and produces gametes, egg and sperm. So when there is a formation of the gametes unite. What happens? The chromosomes get paired. For each trait, we will have a paired alleles. Okay. When gametes form, the paired allele separate randomly so that each gamete receives a copy of one of the 2 alleles. So what happened at the time of formation of gametes? The gametes, suppose this is capital T, small t. So at the time of gamete formation, capital T will move to one gamete and small t will move to the another gamete, right? Though we can have two types of gametes, right? The presence of an allele doesn't promise that the trait will expressed in the individual that it possesses it. In heterozygous individual, the only allele that expressed is the dominant. So we will see in some of the crosses how the heterozygous individual only one allele express itself normally. Only one of the allele express itself, as we have seen in the case of capital P small p, that is the flower color cross example. Capital P from the purple flower we will have a gamete that will be capital P type only, and from the white flower, that is small p, we will have small p type of gametes only. And when there is a union of these gametes, we can have a capital P small p type of plant. That is the heterozygous individual. This is the heterozygous because it is having one capital P and one small p, right? So, Mendel laws of inheritance. Mendel postulated three laws which are now called after its name as a Mendel law of heredity. So, there are laws of dominance and recessive, law of segregation, and law of independent assortment. So, we will see one by one how this laws appear, right? So, the first one, law of dominance. By definition, when two homozygous individual, right, with one or more set of contrasting characters are crossed, the characters that appear in F1 hybrid are dominant characters. So, by taking an example of, again, by this alphabet only, let us solve this. Capital B, that is the purple flower, right? And here small b, that is the white flower. Okay, so they will form a single type of gamete, that will be the capital B, and they will form a single type of gamete, that will be the small b. Now when they are crossed, they will get capital B and small b type of plant only. That is the F1 hybrid, and are shown by the dominant characters and do not appear in the F1 are the recessive characters. So here the capital B is the dominant one, and the small b is the recessive one, So this flower appears as purple, hybrid purple. Right? So I have taken this flower here in this particular clause. Pistil, that is shown by the female plant, that is purple, hybrid purple, and again the male one that will gives you the purple male. So it will have capital B, small b type of genotype, so it will form two gametes, capital B and small b type. The same way, the pistil will be capital B and small b type of gamete, right? So when there is a combination of capital B and capital B, then there will be capital B, that is pure purple flower. And this combination, capital B and small b, then it will be hybrid purple. Now this one, small b and capital B, that will be capital B and small b type, again hybrid purple. Now this one, small b and small b type, it will be white flower, right? So this is showing the law of dominance we can see at F1 generation. This is showing the F2 generation, but this law of dominant we can see at the F1 generation, and in F2 also we can see that this particular capital B small b and capital B small b, this is again showing the dominant character of the purple color that is present at capital B allele. Okay. So let us take one more example. Mendel took tall pea plant, capital T, and the dwarf, small t, and made a cross between them. So here we can see this tall, capital T, and dwarf, small t. Okay, now they are having a cross between them and they all got capital T, small t. That is the hybrid tall. Okay, so in the first generation, he got the result as a dominant tall plant, but this dominant is the hybrid tall. That is the capital T and small t, right? Mendel self-fertilized the F1 generation and obtained 3:1 ratio. So this F1 generation, he taken one male plant and one female plant and made a cross, and he got 3 tall plant, phenotypically 3 tall plant and 1 dwarf plant. So 3:1 ratio is the phenotypic ratio. Okay, but only one of this plant was the pure tall, that is the capital T type, and the two plant were capital T, small t type, and one plant is of small t type, though this is showing genotypic ratio. Okay, so Mendel self-fertilized the F1 generation and obtained 3:1 ratio. He correctly theorized that genes can be paired in 3 different ways, that is capital T, small t, and small t type, right? Now the explanation. The dominance and the recessive of genes can be explained on the basis of enzymatic functions of the gene. Every gene is having some enzymatic function. The dominant genes are capable of synthesizing active polypeptides or the proteins that form functional enzyme, whereas the recessive genes, they are mutant genes and code for incomplete or non-functional polypeptides. So the genes are not functional at that time, right? So the dominant one can express itself and suppress the recessive one. Therefore, the dominant gene produces a specific phenotype while the recessive genes fail to do so. In the heterozygous condition also, the dominant gene is able to express itself. So, at the time of this heterozygous condition, the dominant gene is able to express itself so that the heterozygous and the homozygous individuals have the similar phenotype. So capital T and small t. By appearance, you cannot say that this one is the homozygous and this one is the heterozygous unless you know the genotype of it. Okay. So by phenotypically or morphologically, both the plants will appear similar. Critical appreciation of the law of dominance. So scientists conducted cross-breeding experiment to find out applicability of the law of dominance. So the experiments were conducted by Correns on pea and maize plant, Teschmark on peas, and De Vries on the maize plant. Okay, so these were the 3 scientists who also rediscovered the Mendel law. Okay, so Mendel law work was kept unnoticed for 36 years. When these scientists came and concluded the same law, then they went back and checked whether anyone has done some experiment on the pea. They were amazed to see that 36 years back, Mendel has done all this work and it got published also, but that was unnoticed, right? So this particular work done by the Correns, Teschmark and De Vries were already published by Mendel, right? So Bateson and his collaborators on a variety of organism, by the Davenport on the poultry, by First on rabbits, and Toyma on silk moth, and by many others also have such type of result. They also followed the same results, but the one which was given by the Mendel. These scientists observed that a large number of characters in the various organisms are related as a dominant and recessive. So this dominant and recessive type of relationship which was given by the Mendel, these scientists also, by following on the different plants and different organisms, they also got the same result. Right? So there is an important of this law of dominance. What is that? The phenomena of the dominance is of the practical importance as the harmful recessive character are masked by the normal or dominant character in the hybrid. So in the hybrid situation, what will happen? The harmful recessive characters are masked or suppressed by the dominant one. Okay, so human beings, forms of idiocy, diabetes, hemophilia, are etc. are some of the recessive characters. So that express only when there is a recessive combination, right? A person hybrid for all these character appears perfectly normal. So when the person is having that particular hybrid form, the person will be absolutely normal. It is perfectly normal, right? So thus harmful recessive gene can exist for several generations without expressing themselves. So in that particular human or in any organism, the recessive, when it is in the hybrid form, it will be perfectly appear as a normal, right? But when there is a combination of the homozygous recessive, then it will express itself, right? So the exception of the law of dominance is the incomplete dominance. We will have one more session to discuss this type of exceptions to the law of dominance, that is the incomplete dominance. So after Mendel, several cases were recorded by the scientist where the F1 hybrid exhibited by a blending character of the two parents. So this one showing the incomplete dominance is showing the blending characters. These hybrids were found to be midway between the two parents. So in the blending experiment, what happened? The two will have the two genes or the two alleles will have a different combination. There will be no dominant or recessive type of things. Both will express itself, right? So this is known as incomplete dominance or the blending inheritance. It means that the two genes of the allelomorphic pair are not related as a dominant and recessive, but each of them express itself partially, right? So this happens in the incomplete dominance. Okay, as for example, in 4 o'clock plant Mirabilis jalapa, when plants with a red flower, capital R, crossed with the plant having the white flower, that is the small r, the hybrid F1 will have capital R, small r, and it appears pink flower. It will not appear red, it will not appear white, but it will appear pink. So red is also expressing and white is also expressing and giving a pink appears to the flower. So when these flower F1 plants with pink flower are self-pollinated by, they develop red, RR, pink, capital R, small r, and white, that is small r, flowered plant in the ratio of 1:2:1 in the F2 generation. Okay, so this is all about, we have discussed law of dominance in this particular session. Okay, so this is all about the law of dominance. We will discuss the two other law, that is the law of segregation and independent assortment, in another session. Till then, thank you very much.